Hi everyone, this is Dr. Agalya. Hope you're all doing well. Now we are going to see about the physiology of fertilization and implantation. Before that, we have to also know how the transportation of sperm happens as well as the transportation of ovum happens, right? Okay. So we know this is the testis, right? So testis is the male gonad that is homologous to the ovaries. Testes are suspended in the scrotum by the spermatic cord and it is covered by tunica albuginea. There are around 200 to 300 lobes in the testis and each lobe consists of 2 to 3 seminiferous tubules. Now, so this is the picture. So you are able to see there are testicular lobes and seminiferous tubules inside each lobe, right? So these are the septae that is separating the lobes. This is the tunica albuginea layer, which is the covering of the testis. Then all the seminiferous tubules will join together here to form the reed testis at the apex. Okay. So from the reed testis, the efferent rectangles passes down, which is known as the epididymis, and the epididymis continues into the ductus deferens. Now here in the epididymis is where the maturation of the sperm takes place. Even motility is also occurred here. Now, as the semen is deposited in the vagina, there are around 200 to 300 million sperms deposited, but only a few hundreds of this will get transported to reach the ovum. So, where the sperm is lost? Number one, it is lost in the introitus itself. Second, it is lost due to the digestive enzymes in the vagina act on the sperms. Third one, there are endometrial cells which will engulf the sperms. And fourth one, few sperms are also lost into the peritoneal cavity. As the sperm passes through the female genital tract, it acquires a special capacity to become hypermotile by the influx of calcium ions. So this special property is known as capacitation. Remember, it is just a hypermotility. So the motility of the sperms are occurred in the epididymis, whereas the sperm becomes hypermotile in the female genital tract, and that process is known as capacitation. And this capacitation mostly starts in the cervix itself, but it is more in the fallopian tubes and the uterus. Okay. Now, this is the structure of the fallopian tube. So, we have the interstitium part over here. Okay. So, this is the isthmus, ampulla, infundibulum and the fimbriae. Okay. So, how the transport of the ovum is done? basically what happens is this fimbriae of the fallopian tube swipes over the ovary to take out the oocyte. Okay, and this ovum is transported in the fallopian tube due to the following reasons. Number one, there is ciliary movement of the fallopian tube. Number two, there is fluid that is secreted inside. Number three is due to the muscular contractions of the tube. So basically, if you see the fallopian tube, you will have numerous cilia, okay, and a few secretory cells which secrete fluid, and there are some cells called as PEG cells whose function is not known. Okay, so the cells include PEG cells, secretory cells, and the ciliary cells. And most commonly, fertilization happens in the fallopian tube in the site of ampulla. Why? Because the ampulla is the widest part of the fallopian tube and also it has more number of cilia, right? Now, imagine a sperm has travelled over to reach the fallopian tube, right? And there is also a ovum in the ampulla here. There is also an ovum in the ampulla here. Now, what happens is, we already know as we studied the structure of the follicle, this ovum or the follicle is surrounded by a thickened membrane, that is the zona pellucida, right? So, also there are so many cells that is surrounding the zona pellucida, which is nothing but the cumulus cells. So, in order to meet the oocyte, the sperm has to travel across this cumulus ovaries, then it has to reach the zona pellucida, and then it has to break that zona pellucida and it has to reach the ovum, right? Now, what are the changes that the sperm will undergo at this stage? Then, first state is known as acrosomal reaction. What happens is, here in the acrosome, there are numerous enzymes like hyaluronidase, then hydrolytic enzymes, acrosome, etc. that will help in breaking these cumulus ovaries cells and reaching the zona pellucida for attachment. Okay. And the second one is the cortical reaction. As it reaches over there, the plasma membrane of the ovum and the sperm fuses, and from the cortical granules, numerous enzymes are released further. Then comes the zona reaction. Zona reaction is nothing but basically, as the sperm gets itself attached to the zona pellucida, there are so many glycoprotein ligands that are present in the zona pellucida. The sperm specifically attached to the most specific ligand, that is ZP3 ligand. After that, what happens is, there is thickening of this zona and it basically happens to prevent polyspermy. Okay, so as one sperm reaches its goal, the rest of the sperms are stopped from entering inside. So this is the picture to understand it better. So first, what is happening? The sperm is coming there and making in contact with the egg. Then acrosome reacts with the zona pellucida. There are hydrolytic enzymes in the acrosome. Then acrosome reacts with the perivitellin space. It basically enters the perivitellin space, that is nothing but the space that is present around the oocyte. Okay. So plasma membrane of the egg and the sperm fuse. Then what happens is, as the sperm nucleus is entering the egg, the cortical granules fuse with the plasma membrane and these cortical granules are also released, and then happens the zona reaction, whereas thickening of the zona which prevents polyspermy, right? So, the ligand is ZP3, so that is important. Okay. Now, there is fusion of the female and the male pronucleus to form a zygote. Okay, so this is the zygote basically. Now, formation of the zygote happens within 24 hours. Remember, the fertilization period of ovum is 12 to 24 hours. Fertilization lifespan of sperm is, yes, 48 to 72 hours. Okay. So, within 24 hours, so first there is a fusion of the male and the female zygote, female pronucleus to form a zygote. This happens within 24 hours. Okay. If you take the menstrual cycle, suppose you consider day 14 as a day of ovulation in 28-day menstrual cycle, the most fertilizable days are 6 days before and 3 days after. Okay. Anytime during this time there is a coitus, fertilization can happen. Then we'll see how is the cleavage happening. Basically, in humans, the cleavage is holoblastic, asymmetrical and asynchronous. So, if you ask me, ma'am, what is this? Why do we define cleavage so like this in humans? Because holoblastic, the cell divides completely. Okay? The cell divides completely in the sense it divides completely through the cytoplasm. Asymmetrical, the daughter cells so formed are not uniform. Daughter cells are not uniform. Asynchronous because not all the cells divide at the same time. Okay, so this is how the cleavage happens. First, there is a zygote formed. From the zygote, the division goes like a 2-cell stage, a 4-cell stage, a 8-cell stage, finally a 16-cell stage. Okay. So, this 8-cell to 16-cell stage is specifically called as morula. Okay. So, basically within 24 hours there is formation of zygote. By 3rd day there is formation of this 8-celled morula. Okay. Now, as this stage is formed, what happens is in the morula there is a fluid containing cavity formed. Okay. So, that fluid contained cavity inside the morula is called as a blastocyst. Okay. So, this is a blastocyst where there is a blastocyst, that is nothing but a fluid containing cavity, and you have the cells divided into 2 parts. One is the internal cell mass, another one is the trophoblast. Okay. Basically, a blastocyst is said to have 58 cells. Okay. In that, 53 cells forms trophoblast and the remaining 5 cells form the internal cell mass. So, by day 4, we have the blastocyst formed, and by 4th day what happens is this will start entering the cavity. Cavity is being entered. Okay. Then basically on the 6th to 7th days, implantation starts to happen and it gets completed, and by day 10 to 11 this gets completed. Okay. So, these are the events that will happen. So, this is a picture to understand the process that we have seen till now. So, first what is happening is ovulation is happening, a mature follicle is developed and then happens ovulation and then the fertilization, basically the ampulla of the fallopian tube. Then starts the cleavage. Cleavage is nothing but the division of the zygote cells to form the blastocyst. Then finally, in the state of blastocyst, the implantation happens. Okay. So, this is another picture. So, ovulation is there, then fusion of male and female pronucleus to form a zygote, that is known as fertilization. Then it becomes 2-cell, 4-cell, 8-cell stage and finally a morula that is entering into the cavity, and finally the implantation form is known as blastocyst. Okay. Now, how the endometrium is preparing itself, okay, to receive this blastocyst. So let's imagine this is the uterus. Sorry if my diagram is not good. Try to imagine that this is the uterus, okay. So what happens is, basically, normally the endometrium, imagine this is the uterus. Normally, the inner lining of the uterus is known as endometrium. But as the blastocyst is coming and getting implanted itself, this inner lining of the uterus is now will be called as decidua. Okay? So, this decidua is further divided into decidua basalis. Okay? That is the region where it is getting implanted, and decidua capsularis. So, that region of the decidua that is surrounding the blastocyst upwards. And finally, the rest of the decidua will be called as decidua parietalis. Okay? This decidua basalis and parietalis, okay, say BP, together is known as a functionalis layer, right? And this is further divided into 3 layers, namely stratum, basal, stratum spongiosum, and stratum compactum. Basically, the implantation in human beings is an interstitial implantation. Basically, the implantation in humans is interstitial implantation, and it is a eccentric one. That is, it doesn't implant itself in the center. It implants to either the right corner or the left corner of the body, and it happens in the posterior wall of the uterus. Okay, so it is eccentric implantation. What are the steps in implantation? Number 1 is apposition, number 2 is adhesion, and number 3 is invasion. Okay, so what is apposition? Apposition is basically a loose connection that is happening between the blastocyst and the endometrium. Basically, the endometrial receptive period is between the days 16 to 22 days of a 28-day cycle. Okay, so what happens between these periods is the endometrium will produce a numerous finger-like projections called as pinopods, and this will have some ligand channels. Now, the blastocyst that is coming to get implanted here, right? So, this will have some L-selectin ligands. So, basically, until the period of implantation, there is a loose connection that is happening between these two pinopods and the L-selectin of the blastocyst, and finally, when a time comes, the implantation will happen in the right place. Okay, so that is a loose connection. Next comes adhesions, which is mainly mediated by integrins and cadherins. Okay, then comes invasion, where the blastocyst is now going to bury into the endometrium and it gets implanted itself in an interstitial position. Okay, so what happens is, basically, the endometrial stromal cells are destroyed. Okay, and up to a certain level only this invasion will happen, and after that, due to the presence of maternal immunity, the invasion will be further stopped. Okay, so this is the decidua that got implanted. Okay, so this is the basal decidua, the one surrounding the embryo above, like it's just like a cap, like, so it is a capsular decidua, and the rest of the decidua is known as parietal decidua. As the blastocyst get implanted, there can be some bleeding that can present itself. So that is nothing but the implantation bleeding, and this sign of implantation bleeding is called as Hartmann sign. I hope you would have understood how fertilization and implantation happens. Until we meet in the next video, take care, happy learning, and bye-bye.